In my presentation, I'll talk about CRAWDAD, which stands for Cell Type Relationship Analysis Workflow Done Across Distance. But I think in this field, we try to come up with funny names for our packages, and this is the one we came up with. So first, among the many analyses you can do with spatial omics, one of them is identifying the cell types and the spatial positions they are located in the tissue. So here we have two samples. On the left, you see mouse embryo that it was assayed by Sigfish, and each point is a cell, each color is a cell type. And then on the right, you have a spleen section, a human spleen section, which was assayed by Codex. So this is not transcriptomics, this is proteomics. But in both cases, we can use this information to identify the cell types. And in both organs, you can see, like in both conditions, you can see that the cell types are not randomly distributed in the tissue, but they are highly organized to perform different functions, to communicate with each other, among many things that you can see. So here in the spleen, you have the red and the white pulp. You can see some of the structures here, and in the embryo, you have these different layers of development, and being together in one layer could indicate that this tissue will be together later in development. So the first thing that I wanted to motivate you is that understanding the organization of the tissue is important. Now, what I want to talk about is understanding that the resolution in which we look into this organization is also important. So here we are looking at simulated data. This is a toy example for illustration purposes. But you can see four cell types. Each color is a cell type, each dot is a cell. And here you can see how there are these cells here. There's another cell type in red, there's another cell type in blue. But just look at this style of the image. You cannot come up with any conclusions about the organization in the tissue. But as you zoom out more, you're starting to see that these two cell types are mixed with each other, and there's a cell type surrounding them. And as you zoom out even further, you see that the cell type D, cell type blue, is separated from cell types B and C, and it's separated by cell type A. So using this illustration, I wanted to show that looking into the amount of that you zoom in or you zoom out of the in the image in the tissue, it is important to understand this relationship. Because if you zoom in too much, you won't be able to see anything, but as you zoom out more, you can understand the relationships better. Um, and the third thing that I wanted to motivate you for is it is important to understand the tissue by itself. So looking into the tissue, seeing which cell types are together, co-localized, or separated. But it's also important to perform a comparative analysis. So here we are looking at the human thymus, both assayed by Codex, so proteomics. And we can see that these are both healthy humans, but in later years in life, so the first one is 11 years old, the second one is 21 years old, you start to lose some of the cells in the tissue. And it's important to compare these two conditions, not only to understand what's happening here and here, but also to understand what's the differences between these two conditions, because maybe the cell types organization that's preserved is relevant for a later stage in life. So what I want to present today is CRAWDAD, which is an R package to quantify cell type spatial relationships at multiple scales within and across samples. So here we're looking at spatial organization, considering the scale, so how much you zoom in and zoom out, and we are also able to compare multiple samples. So now I'll dive into how CRAWDAD works. Here again, we have the same simulated data as before, and we are focusing in one cell type. Here we're looking at the reference cell type B. So this is the lime greenish color that we see. And for CRAWDAD, the first step is to draw a neighborhood. So we want to look which cell types are enriched in this neighborhood. So first, we have a neighborhood. We have all the cells from the reference cell type, and we'll draw a circle around those cells. In this process, we define a neighborhood distance, which is a user-defined parameter, and we draw the circle around the cells of the reference cell type. We'll do this for all the cells of the reference cell type. So for all dots that are belong to the B cell type, we'll draw a circle. And then later we create a union of the circles and we define this as our neighborhood. So for example, the neighborhood around the cell type B is this neighborhood highlighted in yellow. Once we define this neighborhood, we'll calculate the cell type proportions. So here, out of all of the cells in this neighborhood, what is the proportion of the cells from cell type B? What is the proportion of the cells of cell type C? And what is the proportion of the cells in the other cell type? So here again, like the figures might not be drawn to scale. Maybe you should have a little more of red here. But you see there are like no cells from cell type D here. And there are a lot of cells from cell type C, a lot of cells from cell type B, and some cells from cell type A. So once we define this proportion, we'll shuffle the data. We need to calculate the new background to compare the proportions to. And the way CRAWDAD define the null background is by dividing the tissue into grids. And these grids will have different sizes. So here we're separating the tissue into grids. Each grid is composed by multiple squares. And the square will have a length scale inside. And once we divided this tissue into grids, we'll shuffle the labels inside each square of the grid. And we're not shuffling the positions of the cells. So here all the points will have the same X, Y position after shuffling. We're just shuffling the labels. And we shuffle the labels inside each grid. So for example, a label that was here can go here, but a label that was here cannot go to this other grid. So this is not happening, this is happening. And the idea that we're trying to aim here is like to create a new background by controlling the length scale. And essentially what we're doing at this point is trying to look at these multiple scales by creating multiple new backgrounds at the multiple length scales. Once we shuffle the data, we have our new distribution for this particular length scale. And then using the same neighborhood as before, we're not changing the neighborhood, we're using the same one as before. We'll calculate a new proportion. And once we calculate in this new proportion, we see that cell type D increased, cell type B and C decreased, and cell type A increased. So now, with this information, we'll compare the proportion we had before to the proportion we have after shuffling the data. And using a z-test, we'll calculate the z-score using this two proportions. So proportion 1 is the one before the original proportion. Proportion 2 is the proportion after shuffling. And here what we are comparing is how much the proportion after shuffling is different than the original proportion. And we'll calculate a z-score, but we repeat this process for multiple length scales. So we'll define a length scale, we define a size for the square, and then we calculate, and then we increase, calculate the z-score again, increase, calculate the z-score again, and do on and so forth. So you see one of the graphs that we have for this type of data. But we also do the grid permutation. Another type of permutation that we do, so it is important to clarify, is changing the grid, because sometimes if we have a particular structure in the tissue and by chance our grid matches the pattern in the tissue matches our grid size here, all the labels we shuffle only inside the grid, so the proportion before and after won't change much because this grid is composed mostly by the same two cell types. So we also perform other shufflings in the data, so we try to change not only the size of the squares, but also the initial starting points of the square. So if you look here, the original starting point was here, and then we moved a little bit to the left, a little bit to the top, and then repeat this process again, now moving left and to the top again. So we try to accommodate um, for these caveats that can come for whenever your grid matches the pattern in your tissue. And once everything is done, once you repeated this process for all the permutations in grid and all the permutations in length scale, you come up with a z-score trend. So here what we have on the y-axis is the z-score, on the x-axis is the scale, and for each scale, which is the size of the squares that we use to shuffle the data, you have a z-score. And here you can see the error bars. The error bars are from the grid shuffling. Whenever we switch the X and Y positions of the grid. And we also have a significance threshold. So by calculating these multiple z-scores, we can see that how this relationship changes once we changes the scales. And we define colocalization as we define that cell type B is enriched in the neighborhood of reference cell type C whenever the z-score crosses the threshold. So here it's around 250 micrometers. And for this other graph that we have, we can see that both of them are enriched, this relationship happens at a greater scale. So here we are looking at maybe 100, 600 micrometers, which means just by looking in the pictures that cell type B and C are intermixed together, while cell type A, although it's next to cell type B, is not as mixed with the cell type as cell type C is. So you need to zoom out a little bit more to identify this relationship. So this is how you can interpret CRAWDAD results. And you will have this trend for each cell type pair. The relationships are not symmetrical. So using reference cell type B and neighbor cell type C won't produce the same results as using reference cell type C and neighbor cell type B. But we define colocalization whenever there's a mutual symmetrical enrichment of one cell type in the neighborhood of the other, and we define separation whenever there's a depletion, a mutual depletion of one cell type in the neighborhood of the other. And to summarize all these informations, because we would have a trend for each cell type pair, we created a dot plot. So here we are summarizing just the point in which the trend crosses the significance threshold. So here for cell type C as reference and cell type B as neighbor, we have a big red dot, and the red indicates a positive z-score, which is for colocalizations. And the size of the dot represents the scale in which this relationship happens. And we are trying to focus more on relationships that happen at a finer scale. So bigger dots will represent finer scales, smaller dots will represent greater scales. And these other relationships that we have for A and B happens at a greater scale is still a positive z-score. So you see a smaller red dot here. And with now I explain how CRAWDAD works, so I'd like to present some data, some results in real data, not simulated data. And here we're looking at the mouse cerebellum assayed by slide-seq. So here we're focusing three cell types, the Berman glia, the oligodendrocytes, and the Purkinje cells. And the Berman glia and the Purkinje cells, they are in the same cerebellum layer, while the oligodendrocytes are in a separate cerebellum layer. So looking at the relationships with the stars that we're used to highlight, we can see that the Purkinje cells are colocalized with the Berman cells because both are enriched in the neighborhood of each other, while they're separated from the oligodendrocytes. So this is what CRAWDAD identified. And once we look into this relationship in the real tissue, we see that the Purkinje is intermixed with the Berman in their own cerebellum layer, while they are separated from the oligodendrocytes, which belong to another cerebellum layer. Now, looking at tissues that are not as organized as the cerebellum, here we're looking at a human breast cancer assayed by xenium, and we can see that even though it's not as organized as the cerebellum, there is still some structure that was identified by CRAWDAD. So here we have the in situ carcinoma and the cell types that share a similar result. So basically what we did here is we performed the dot plot, calculated the relationships for all the cell types paired, then we perform hierarchical cluster just to organize the cell types, and we are able to locate the cell types that share a similar colocalization or separation pattern. And using this, we can locate like the different tissue structures. This method is not defined for identify tissue structures, but you can identify the cell types that have a similar organizational pattern. So here you have the in situ carcinoma. You can also have like the invasive tumor, and you also have like the other cell types, including a mixture of immune cell types that form similar that have similar spatial relationships, and you also have cell types that pretty much don't have any spatial relationship. Um, CRAWDAD was not the first method to look into spatial organization. There are other methods. So the first one is an older method, but is very popular. It's called replace-K. It is used initially, I think, for geospatial analysis, but it can also be used for spatial omics. And I just want to highlight the difference between CRAWDAD and replace-K. So in replace-K, you start with a neighborhood from a particular reference cell type. So in this sense, it's similar to CRAWDAD, but in replace, you increase your neighborhood. So start with smaller circles, and then you increase the radius of the circles, and then you calculate the proportion of or the counts of the other cell types that are inside the circles, and you compare it with the global proportion. So in replace, you're increasing the neighborhood size and you're always comparing with the global proportion. In case of CRAWDAD, your neighborhood is always the same, and you're all trying to accommodate for the difference in length scales by changing how you shuffle the new background. So by creating grids of different sizes, um, you create new backgrounds that were shuffling like in different length scales, and that's how you compare, like you analyze the different scales. Here, you're just increasing the neighborhood size, but you are always comparing with the global density. Another method that was implemented by SquidPy, but it was initially developed by Toasty et al. It's the co-occurrence analysis. And in this case, you're also increasing the neighborhood size, but in this particular method, they're trying to look for annulus regions, or here in the US, people eat a lot of donuts, so it's donut-shaped regions. Um, and then you have this neighborhood, but the neighborhood is just this gray part that I'm highlighted here. So you start with this neighborhood, and then you increase the neighborhood size, but you're always looking in this annulus regions that you find here, and you're not considering the other parts. And the statistics a little different, but you can always think of like increasing the neighborhood as you do in replace and comparing to like what is globally available there. And how this results like impact the analysis. So here we're also looking into the cerebellum, same data as you saw before, looking into the Purkinje cells with the oligodendrocytes and the Bermanglia. So we saw that Purkinje is separated from oligodendrocytes and colocalized with the Bermanglia. And you can see this relationship happens in CRAWDAD at like at a very early scale. And for Ripley's, you can see that it happens in the beginning. Uh, there's like no threshold that you can use to identify this, but then at later scales, once you increase Ripley's more, you see the opposite. Uh, and then you see like a swift, like a change in this relationship trends. And the same is true for for the squid pie co-occurrence. Initially, you see there's like a higher score for the Baymanglia, but then after a certain scale, they switch and they switch again. And I'm not here to claim the other methods are wrong. I think it is just a definition of the neighborhood. So here you're using an annulus region for the neighborhood, and that can change a lot of your results. But depending on the analysis you're trying to do, this might be the better, like a most suitable method. But looking into this like this analysis in this particular example, CRAWDAD was better, was able to identify these relationships better than the other cell types. We also performed an analysis in a null simulation. So we simulated Gaussian random fields. So each point has a value, and it can be a high value or a null value. And then we binarize this data. So you can see that like high values will be associated with one cell type, low values will be associated with other cell types. And in this case here, we have four cell types. And if the cell types came from the same simulation, they should be separated from each other. If they came from distinct simulations, so here we have two simulations, they should have not any significant relationship with each other. And when we performed this over like 100 simulations, I think, or 10 simulations, we saw that CRAWDAD was able to had a higher true positive rates than the other two methods. Um, But this so far I talked about the analyzing samples individually. I also want to mention that CRAWDAD is able to compare samples. So here we are looking into 9 mouse brain samples assayed by Murfish. And here we have 3 replicates from, so 3 different mice, and we have 3 different locations in the bank. So the first, we section the brains from a further, like more frontal locations, then from the middle, and then from the back. And these are location 1, location 2, location 3, not necessarily in this order, but then we have 3 different mice. So what we do with CRAWDAD, we will calculate the z-score trends for all cell types pairs. So imagine here you have a cell type pair, and then you calculate the z-score trend for that cell type, one as a reference, other as a neighbor. And in order to compare the multiple cell types, we needed to come up with an idea to how to summarize this information. And the way we came with this idea, there were many other ways of doing it, but what we did was calculating the area under the curve. So here we have the area under the curve for this particular cell type pair. So if it is a colocalization, we have a sine area under the curve, so it will be a positive area under the curve. If we have a negative relationship, we'll have a negative area under the curve. And using this information, we came up with a matrix in which the columns will be the area under the curve for the particular cell type pair. So here we have the columns, which is the area under the curves. And here we'll have the samples. So for example, for this sample here, location 1, we replicate 3. We'll have an area of the curve for relationship of astrocytes and inhibitory neurons. And then we have another one for astrocytes and microglia, and then so on and so forth. So using this matrix, this is very similar to single cell analysis or any other like gene expression analysis because you have samples and you have features for those samples. So one way of doing this is by calculating, uh, by performing the principal component analysis, and we perform principal component analysis on this matrix. And we notice that samples that came from the same location are more similar than samples that came from different locations. So this is what we're expecting. And we also are able to dive in further into these results by analyzing the variance of these relationships. So here we are looking into the variance across replicates. So looking into the same location, so tissues like tissue slices that were sectioned from the same location of the brain. And here we are comparing locations within the same mouse. We are able to identify which cell types varied the most, and then we visualize the z-score trends for those cell types. So here looking at GABAergic neurons and excitatory neurons, and then we see that if they come from the same location uh, of the brain, they all have like a negative area under the curve, and they all have z-score trends that are negative. But depending on the location of the brain that you get, you might even have positive z-scores. And we visualize this in space and notice that there are different patterns. So depending on the location that you get, for example, for this location here, you can see a very central pattern. For this location here, you can see the GABAergic neurons are more dispersed in like the central side of the tissue. So this is good to show that CRAWDAD works for comparing samples. We also analyze multiple human spleen samples, and I'd like to thank our collaborators for collecting this data for us. And we are able to identify the cell types that are in the red and the white pulp of the spleen, but we are also able to compare these multiple human spleen sections and see that for some cell type pairs, you see a consistency in within like patients. So here we have 6 samples and 2 samples per patient. So we have 3 patients. Each patient is a color, and each line is a sample. And we see that for Ki67 proliferating cells and follicular B cells, this patient in yellow had a positive Z-score. So this cell types are co-localized in that particular patient. While for other patients, the one in blue, we had a negative z-score, meaning that for the particular patient, there was a separation of these two cell types. But also, these relationships are not always consistent across patients. So for example, here we have a positive z-score trend for one of the samples, and here we have a negative z-score trend for another of the samples coming from the same patient, which could be from technical artifacts. It's hard to know, but in some of these cases we can see consistency across patients. CRAWDAD uses square grids, but we can also use hexagonal grids for shuffling the data. We see there's a correlation of the z-scores regardless of you use whenever you use hexagon or if you use square grids, still very comparable. One of the things that I want to highlight is I mentioned before the results are not always symmetric. So in this case, we have two cell types. We have granule cells and UBC cells. And here first we have the neighborhood of the UBC cells. The UBCs are very sparse, and therefore you can see the neighborhood there. There are not a lot of cells from this particular cell type. But for all the neighborhood, you can see there's a higher presence of granule cells in this case. And then once you look into the neighborhood of the granule cells, which you see here in the right, uh, out of all the cell types there, because the UBC cells are more sparse or more rare, um, they do not significantly contribute to the proportion uh, of the UBCs in that neighborhood. So you can see that basically, in other words, in more simple terms, whenever you see UBCs, you see granule cells. This is what's showing here. But whenever you see granule cells, you might not see UBCs. So that's why there's a asymmetry in these relationships. And here's another example. You can see that the prosemitic mesoderm cells are mostly here. There's a high density here in this area, but there are still some other sparse cells in other regions of the tissue, and they contribute for the creation of the neighborhood, but they don't contribute so much for the proportion of the other cell types. So you can see that these relationships are different. Um, they are not symmetric for this particular case either. Um, And I think by now, I hope that I have motivated you to why it's important to understand cell type spatial relationship and also why it's important to understand it in multiple scales. Um, it is also important to do a comparative analysis. So I'm not looking into a single sample, but to compare samples to see what's happening in one condition versus the other. CRAWDAD is an open source R package, so you can download it from GitHub. It's applicable to any spatial omics technology with cell types and positions. So whether you have spatial proteomics with Codex or whether you have spatial transcriptomics with Murfish, Xenion, as long as you have the locations of the single cells and the cell types annotated, you can apply CRAWDAD. I would like to thank my professor, Jean Fenn, for advising me and all my lab for the support. Thank you very much.